Transformation observed by Griffith. In Streptococcus pneumoniae. In this, it is one bacterium, genetic material present. It is another bacterium, genetic material also present. 1 and 2. This bacterium dies. By any way, by any method, it dies. Now, genetic material from this bacterium comes out into the medium, surrounding medium. There is either water or soil or blood, whatever it may be. Now, this genetic material that attached to the recipient bacterium, one of the strand hydrolyzes, remaining strand that enters into the bacterium with specific proteins and combines with the main genetic material in inheritable form. That is called transformation. This experiment first carried out by Griffith in Streptococcus pneumoniae. It is the basis for the discovery of the genetic material is the DNA. Griffith experiment, that the transformation experiment is the basis for the discovery of the DNA is the genetic material. So, through the medium, the DNA of one bacterium that enters into the another bacterium in inheritable form, that is called transformation. And next and last, transduction. In this, it is one bacterium, it is another bacterium, genetic material. Virus which attack one bacterium, it carry the genetic material of that bacterium to the another bacterium. So, if genetic recombination occur or transformation of genetic material carried out by virus, that is called transduction. How this transduction carried out by this virus? We will discuss that in replication of virus. In virus, replication of virus, that is the lysogenic cycle of virus involved in transduction. So, transduction, transfer of genetic material with the help of virus, by virus, from one bacterium to other bacterium, transduction. It observed by Lederberg and Gender in Salmonella typhimurium. By any method, either conjugation or transformation or transduction, new genetic material that added to the cell, that is called genetic recombination.